On behalf of the Undiagnosed Disease Network and the Undiagnosed Disease Network Foundation, we are happy to welcome you to our webinar on advancing hope and breakthroughs in undiagnosed and ultra-rare diseases. I am Christine McGarvey. I am a rare disease advocate, a UDNF peer member. I'm also a UDN participant and a mother of a UDN participant. I'm joined here with Dr. Sessions Coles, a physician with a long-standing interest in finding diagnosis for undiagnosed individuals with unusual medical problems. This webinar will highlight the work of the Undiagnosed Disease Network, whose mission is to solve medical mysteries through team science, and the Undiagnosed Disease Network Foundation, whose mission is to advocate for research that advances diagnosis and treatment for undiagnosed individuals. Today, more than 30 million individuals are living with medical conditions that do not have diagnosis or are ultra rare. Approximately half of these individuals are children, and the average time from symptom onset to diagnosis is about 7 years. I myself am over a decade in my diagnostic odyssey, and this year my daughter is at her 7-year mark. And fewer than 5% have specific therapies that are known to help. Despite the scope and impact of the problems posed by undiagnosed and ultra-rare disease communities on undiagnosed individuals and their families, the providers who search for diagnoses, health insurance companies, employers, and society research to find the best practices for diagnosis, care, and therapies is lacking. Our webinar today will describe both diagnosed and undiagnosed cases, outline strategies to identify therapies once diagnoses are established, and describe the research priorities of the undiagnosed and ultra-rare disease communities. We will start by highlighting the history and the achievements of the UDN and the UDNF. The UDN was made possible by the completion of the Human Genome Project in 2004, which provided a blueprint for the human genome and the vision of a geneticist and researcher at the National Institutes of Health, Dr. William Gull. Dr. Gull recognized that although the human genome includes about 20,000 genes, gene code sequences differences in only about 5,000 of the genes were known to cause disease. He hypothesized that using new technologies for gene coding sequencing developed by the Human Genome Project and new computer analysis strategies, he and his colleagues combined careful clinical characterization of undiagnosed individuals with an unbiased search in all 20,000 genes of their human genomes. This search focused on discovering differences in gene code sequence between unaffected and affected individuals that might account for their symptoms. He started the Undiagnosed Disease Program at the NIH in 2008. It was successful, and thanks to the support from Dr. Francis Collins, the director of the Human Genome Project, and later the director of the NIH, the Undiagnosed Disease Network was established in 2014. The original UDN included 7 sites at which undiagnosed individuals were evaluated, a sequencing center where DNA sequencing was performed for all accepted UDN participants, and core laboratories that included model organism screening core, which used model organisms such as fruit flies, worms, and fish to help establish whether discovered gene code differences were in fact causing an individual's disease. Because the UDN was established as a research network focused on developing best practices for diagnosing undiagnosed individuals, individuals applied to the UDN, and if they were felt to have a set of medical conditions and symptoms that were likely to be diagnosed with UDN resources, they were accepted and assigned to one of the clinical sites for evaluation. In addition to an in-depth clinical evaluation that included whatever the medical conditions and symptoms of the undiagnosed individual required, for example, state-of-the-art imaging, specific blood tests, and evaluation by multiple subspecialists, the UDN participants also received whole exome and later whole genome sequencing Frequently, this sequencing involved not only the affected individual, but also his or her or their first-degree relatives, usually parents or sibling. In my family's case, both me and my daughter, and also my mom, my sister, and my husband had their genome sequenced. This family-based sequencing strategy made the computational identification of gene code differences that might be causing the medical conditions and problems more straightforward. Remember that all of us carry more than 1 million gene code differences, most of which do not cause disease. Finding the causative gene code difference is like searching for a needle in the haystack. Next, Dr. Cole will discuss the expansion of the UDN. Dr. Cole received his medical degree from Yale University School of Medicine, and he received his pediatric and neonatal perinatal medicine training at Boston Children's Hospital at the Harvard Joint Program in Neonatology. He served on the Harvard Medical School faculty from 1981 to 1986, and he joined the faculty at Washington University School of Medicine in 1986, where he held a variety of clinical and administrative leadership positions until his transition to emeritus status in June of 2024. He served as a co-principal investigator for the UDN's Clinical Research Support Corps and its Data Management Coordinating Center, and currently serves as a consultant for the UDN's DMCC at Harvard Medical School. He also leads the UDN's Therapeutic Matching Committee aimed at identifying therapies for ultra-rare disease patients, and Implementation Science Advisory Board focused on improving access to UDN best practices. He has served as secretary and member of the executive committee of the Undiagnosed Diseases Network Foundation since its establishment in 2022. He has introduced, advocated for, and supported family-centered care in the NICU and for undiagnosed and ultra-rare patients and families, a strategy that includes and empowers families through full partnership in medical care decisions.
Thank you, Christine. Because of the success of the UDN, the NIH expanded the network to 12 clinical sites in 2018 and integrated more advanced methods for DNA diagnosis. In addition, because impact of gene code differences on gene function, some gene code differences cause genes to be overactive, some gene code differences cause genes to be underactive. Because these differences in gene function may leave specific, unique signatures in blood, urine, or other bodily fluids, the UDN created a metabolomics core that specializes in state-of-the-art methods for detection of these unique differences in metabolomic signatures. Finally, the UDN also launched a biorepository to which UDN investigators would send blood, urine, or other samples from individual UDN participants to ensure their availability for further research within and outside the UDN. From 2019 to 2024, the UDN has focused on expanding its state-of-the-art strategy for discovery of genetic causes of undiagnosed diseases by using different methods for sequencing called long-read sequencing and epigenomic sequencing. In addition, the UDN has expanded its data sharing platforms to optimize availability of its discoveries within and outside the UDN, and has initiated a group called the Therapeutic Matching Committee that leverages the functional characterization of the diagnoses made in UDN participants to help identify therapeutic strategies that would work for individuals and might be pursued. The research of the UDN and, more recently, the support of the UDNF have established best practices for diagnosis of undiagnosed individuals. The secret sauce that the UDN uses to shorten prolonged diagnostic odysseys is the integration of multiple experts who each contribute to helping with diagnosis. Historically, these experts have pursued their research in silos. However, the UDN engages leading experts in genomic sequencing, experts in establishing individualized metabolomic, epigenetic, genomic, and transcriptomic signatures associated with likely disease-causing genomic variants, a so-called multi-omics approach, experts in strategies to test the impact of likely disease-causing variants in model organisms, as mentioned by Christine, fruit flies, worms, and fish, and experts in development of individualized precision medicine approaches for caring for undiagnosed individuals along with exceptional clinicians. This team is then enhanced by using the internet-based matchmaker exchange to identify additional experts outside the UDN who have interest and expertise in genes and methods not represented within the UDN. Established in 2021, the UDNF has added to the UDN's impact by developing plans for sustainability of the UDN when funding from the NIH expires in 2028, by advocating for additional research support for the UDN, and by ensuring that individuals and families affected by undiagnosed and ultra-rare diseases are fully represented and engaged in all individual and network-wide discussions. The results of these efforts are described on the next slide. Despite accepting only the most difficult cases, the UDN has achieved a remarkable 29% diagnostic success rate. This success rate includes discovery of 77 new diseases that were previously unrecognized by medical doctors. While some of these diagnoses could be established by direct clinical testing, for example, a specific blood test or imaging study, most of these diagnoses were established through whole exome or whole genome sequencing. The symptoms of 49% of UDN participants are neurological in nature.
Thank you so much, Dr. Cole, for going over the technologies utilized within the UDN and for highlighting the research efforts that the UDN has accomplished this year. We hope that this brief introduction to the UDN and the UDNF has highlighted the extraordinary work of both organizations. Both are critical contributors to the progress for the undiagnosed and ultra-rare disease communities. To illustrate the strategies and impact of the UDN and the UDNF, we will now turn to case studies of undiagnosed and diagnosed UDN participants presented and discussed by UDN and UDNF members. These case studies will help highlight the importance of research priorities, approaches, achievements, and challenges to ensure continued progress. Finally, Dr. Meghan Halley, the chair of the UDNF Board of Directors, will summarize key points from the webinar, emphasize the importance of ongoing research, data sharing, and community support, and encourage your participation through donations, advocacy, and involvement in UDNF activities, and announce the upcoming events and the end-of-year campaign to motivate continued support. We look forward to the day when individuals affected by undiagnosed and ultra-rare diseases can all access the best practices discovered and tested by the UDN and the UDNF. Our diagnosed case study will be presented by Dr. Hugo Bellen, Dr. Robert Bucelli, and Michele Herndon. Dr. Hugo Bellen is a distinguished service professor at the Baylor College of Medicine in the departments of molecular and human genetics and neuroscience. He received his PhD in genetics from the University of California at Davis and completed postdoctoral research in the laboratory of Dr. Walter Gehring at the University of Basel in Switzerland. He was an HHMI investigator at Baylor College of Medicine from 1989 to 2021, and he joined the Neurological Research Institute at Texas Children's Hospital at its inception in 2011. Dr. Robert Bucelli graduated from the MSTP program at the State University of New York at Buffalo in 2007. He trained in neurology and neuromuscular medicine at Barnes-Jewish Hospital and Washington University School of Medicine. He then joined the faculty at the Washington University School of Medicine in 2012 as a member of the neuromuscular section of the Department of Neurology. In addition to having an active clinical practice and performing electrodiagnostic testing, Dr. Bucelli also co-directs the Washington University School of Medicine ALS Center and serves as Associate Director in their clinical laboratory, where he functions as a nerve and muscle pathologist and oversees the laboratory's biomarker testing. He has been involved with the Washington University School of Medicine UDN site as a participating clinician since the site's launch and was added as a site co-investigator in 2023. In addition to ongoing clinical support for the UDN, he also sits on the site's case evaluation committee. Last, we have Michele Herndon, who is the program director for the UDNF Patient Navigator program, and she is also the co-founder and vice president of the Mitchell and Friends Foundation.
So hi, I'm Bob Bucelli. I'm a neurologist at Washington University School of Medicine in St. Louis. It's a real pleasure and privilege to be able to talk to this group today about Mitchell syndrome, or ACOX-1 gain of function. I'll be talking a little bit about the clinical features of this disorder and the journey that our index patient went through to get this diagnosis, and I'll be accompanied by colleagues Hugo Bellen and Michele Herndon to follow up. These are my disclosures, and I thought it was important to just mention the fact that I am on the Medical and Scientific Advisory Board for the Mitchell and Friends Foundation, which you'll likely hear more about, and then I'm the PI for the Mitchell Syndrome Fund here at Washington University in St. Louis. So this is, I've talked about this disorder in different venues before, and this is the most condensed I've ever gotten with talking about this just incredible individual. I put an 8-year history that he went through with his family on one slide. So our index patient started off as a healthy 12-year-old boy and came to medical attention here in St. Louis with new gait issues, hearing loss, and eye symptoms, almost like a foreign body sensation in the eye. And in the course of the evaluation was found to have a very longitudinally extensive spinal cord lesion that predominantly was located in the dorsal columns and extended throughout the cervical thoracic spinal cord. In addition to this, there was peripheral nervous system involvement manifesting with a sensory ataxic neuropathy and lower limb weakness. And appropriately so, he underwent very extensive comprehensive evaluations, initially locally and then without answers, sought out care nationally, and no definitive diagnosis had been made. And because of the gravity of what he was dealing with, he, again, after very, very thorough evaluations at multiple institutions, he was treated quite aggressively with empiric aggressive immunomodulatory therapies for what was characterized as a syndrome of myeloneuropathy with hearing loss, but the cause was not clear for the majority of his clinical course. And again, to summarize a lot on one slide, there was a waxing and waning course to his illness. During some periods, he would decline and have a precipitous decline in certain points and then recover, oftentimes in the setting of being treated quite aggressively. But in retrospect, when you look at the overall clinical course, there was kind of steady background progression. And 7 years into his clinical course, by the age of 19, he had progressed to being wheelchair bound with dense lower limb deficits and near paraplegia. Had required cochlear implants at that time to address his hearing loss. And around this time was admitted with respiratory failure due to his neurologic disease, and this culminated in need for a tracheostomy. Ultimately developed an encephalopathy, and given the involvement of the brain, and I can't go into the details behind this in the interest of time, but this is his most recent brain MRI before he decompensated. Again, there's artifact from the cochlear implants here, but the white matter was okay at this time. And then again, within a month, there was progression to involve the cerebrum. And given lots of discussions with both directly the patient and his family. He opted to move to comfort measures at that time. And he, in a very, very selfless way, and this was kind of a defining attribute of Mitchell, he donated tissue to our postmortem donation program, really with the goal of advancing the understanding of his condition. And we've learned a ton already, and we'll continue to learn from these donations. This is showing a correlate for what we saw in imaging. So this is a stain that stains axons, and the brown staining is axons. And this is actually the portion of the spinal cord that we saw that was white on that original imaging, where there's really complete axon loss in the setting of his condition, the spinal cord. And then this is up in the cerebrum, the area that was involved more recently, and it has a very typical finding on this LFB-PAS stain, showing pallor underneath the cortex, which is with an area of what we call U fiber sparing. So it's almost a demyelinating or dysmyelinating pattern, sparing the area right underneath the cortex, which is very common in disorders, paroxysmal disorders and other disorders that you'll likely hear more about in later slides. These past slides were just provided by Nima Sharifai and Sonika Daria here at WashU, so I wanted to acknowledge them. So during the time that Mitchell was undergoing this extensive odyssey, he, in 2017, this is a direct quote from his mom, Michele. She recalls him saying, "Mom, I don't care if they ever find out what it is. I just want to get better." He, at this point in time, in 2017, our site, Washington University in St. Louis, wasn't a clinical site established yet, and he went down to Baylor at the suggestion of his rheumatologist, Tiffany Vogel, and was seen by colleagues at Baylor. And you'll hear more from Hugo Bellen here, pictured in the top right. Him and Lok Chung really made the major breakthrough with respect to Mitchell's case through the UDN. This was the first time that I really had any exposure in the clinical realm to ACOX-1. You're going to hear a lot more about this protein and its function. But to summarize, it's the first and rate-limiting enzyme in very long-chain fatty acid beta oxidation. This occurs in peroxisomes, and a product of its function is peroxide production. And they, through their work at Baylor, ended up finding that Mitchell had a de novo variant in this gene that you'll hear more about. And this will be the first time you'll hear this, but this specific variant resulting in a protein change of N237S has really become a signature of what is now known as Mitchell syndrome. So this is the original paper from Neuron in 2020, and I've just highlighted the fact that Mitchell, to Hugo's credit, Mitchell was included as an author on this, just given all of the contributions that he made to describing this disorder. And soon thereafter, with describing this clinical syndrome, there was a kind of cascade in the clinical literature of additional patients being described with a very strikingly similar phenotype, and all around the world and in the United States. And this is just a kind of highlight of kind of the pace and rapidity at which new discoveries have been made and new recognition of cases have been made since those early days in 2019, 2020. So these are the three patients. And the details of the slide aren't as important as just kind of the numbers. So these are the three original patients that were described in the original manuscript that I've referenced already. And Tiffany Vogel helped us put this together. So again, Mitchell, 2017, when he was evaluated by the UDN as patient 1. And again, first three patients, 2020. By 2023, there had been 15 patients identified with this condition, and it's very clear that there was a very striking, distinct phenotype associated with this particular variant, the N237S variant in ACOX-1. And a group of individuals, again, Tiffany Vogel at Baylor helped kick this off, but this group of individuals, I don't have time to go into all their names, you'll recognize one of them, here's Michele Herndon, has really banded together here at WashU to start to work on a retrospective natural history study. And so to date, we know of about 31 patients worldwide with this condition. Again, all to date have been confirmed to have that specific variant in ACOX-1, all very typical phenotypes with some variability. And this is kind of where we're at right now, present day live data at WashU with describing this clinical condition with 20 patients in our retrospective natural history study that's really being led by the individuals on the top of this slide. On the translational side for work in therapeutics, we've been working, and you'll hear more about NACA as a potential therapeutic for this condition. We've been working for many years now with industry to try and pursue an investigational new drug application at the FDA, and that is now reality and ready to go when the time is appropriate for a patient. We've been using things like vitamin B2 or riboflavin with some anecdotal clinical success. And others around the world and country are also working on this. So this is generation of a zebrafish model. And I mentioned Lok Chung, and he's continuing to work on this at the University of Houston and continues to collaborate with the Mitchell and Friends Foundation present day and is actively working on this front as well. And here at WashU, there's been a big initiative to try and partner with folks at the ALS Center, which I co-direct. And a lot of this work is being done by individuals I'll mention later, but the basic idea is you'll hear more from Hugo about animal models of this condition. And the big question, of course, like it always is in translational medicine, are the findings that we're seeing in animal models applicable and translatable into humans? And so this is the big focus here. We're using fibroblasts and induced pluripotent stem cells from affected patients that have been evaluated here, and investigating them on a number of different levels to not only better understand the disorder, but also in the hopes of testing and rapidly testing different therapeutics. And so this is work or a slide from Zito Hubler, who's a postdoc in Tim Miller's lab, who's working on this, and Kathleen Schuck, who's junior faculty and a collaborator of Tim's. This team has really done a lot of incredible work already, taking this approach of using a human cell model. And the goal really, again, is to increase understanding. And in parallel with that, as understanding increases, the hope will be to identify more meaningful therapeutics in a very intriguing area of potential therapeutic intervention for this disorder. And appropriately, having this being done in Tim Miller's lab is the idea of developing antisense oligonucleotides to block the gain of function that we're seeing in this particular with this particular change in the ACOX-1 gene.
Well, upon this nice introduction from Bob, I'd like to talk about the work that we've done that relates to Mitchell syndrome. And the key take-home message here is that in Mitchell syndrome, we see elevated levels of reactive oxygen species, which I'll elaborate on. Much of this work was done by Lok Chung, who's currently an assistant professor at the Methodist Hospital, and the program was started by Tiffany Vogel, and who kind of brought this to the Undiagnosed Diseases Network. And you just heard from Bob Bucelli about the phenotypes that were associated with Mitchell. So the protein that's encoded by the ACOX-1 gene is a peroxisomal enzyme and is involved in the beta oxidation of the very long chain fatty acids. I need to explain this a little bit. Peroxisomes are little organelles that are present in most of our cells. And one of their main roles is to kind of take very long chain fatty acids that are typically, for example, found in myelin, and to bring them through a transporter in the peroxisomes where they're degraded by this ACOX-1 protein. That is, they're shortened by the ACOX-1 protein. And this process of shortening the very long chain fatty acids leads to the production of high levels of H2O2. And that is a toxic reagent in the sense that H2O2 can damage lipids, proteins, DNA, and therefore needs to be removed and turned back into water. And that is done by an enzyme called catalase. So the shortening of the very long chain fatty acids in the peroxisomes is critical because then they can be used as an energy source in the mitochondria in the cells. So the process in human is being already associated with other human disease. For example, there's mutations in this ABCD1 transporter, which cause X-linked adrenoleukodystrophy. And so this is the function of peroxisomes is not weird to medicine. It's been implicated in other disease already. So what Locke did was to first look at this patient mutation, which is, as Bob mentioned, a single amino acid change. And if you take this protein and express it in fly cells, what you notice is that if you express the wild type, the normal protein, you see that indeed this protein co-localizes with the peroxisomes, just to make sure that in flies, the same process is happening. And when you overexpress this mutant protein that's found in Mitchell, you see that there is a lot more of this protein present in these peroxisomes. And what turns out to be the case is that when you probe for the level of this protein, you use the same drivers here, so you should have the same expression level, but you don't. And when you look at a Western blot and look at the protein, you see that there is a lot more protein in the cells that express the Mitchell mutation than in the normal cells that express the wild type protein. And it turns out that there is an increase in the level of protein, and there is an increase in what we call a dimer. Normally, this protein functions as a dimer, and that is what is shown in the next slide here. You have a monomer, you have this point mutation, and this is inactive. And when it forms in the presence of this substrate FAD, an active dimer, it'll produce this H2O2 and reduce the very long chain fatty acids to the long chain fatty acids. And it turns out that this single amino acid change in this protein leads to an active protein, an active dimer that is constitutively active and is always producing H2O2. And so this is important because you saw that Mitchell, based on the previous presentation, had problems in the nerves, and the nerves are insulated. Each of the nerves or axons is insulated by myelin. And these layers of myelin are like, if this were an electric cable, the cable is insulated by plastics. And you can think of as myelin as the plastic that insulates the iron cable, which is important because you need to insulate and make sure that when there is electrical activity, it's not spilling over to neighboring cells. And it turns out that the peroxisomes are very important in these myelinated cells, in these cells that protect these axons of these neurons. In fact, they provide a whole series of functions that relate to membrane stiffness and allow membranes to interact. And they're called sphingolipids, and there is a whole variety of these sphingolipids. But it turns out that there's a very high level of these very long chain fatty acids in these myelinated cells. And so based on the work that was done at WashU with Dr. Bucelli and his colleagues, he sent us this slide and showed that in the patient, you see a problem with this insulation. Normally in the in normal individuals, you see these beautiful cables that are each insulated by myelin, and this was not the case in some of the biopsies taken from Mitchell. And if you look carefully, you can see that there is less myelin and there is a progressive demyelination. And that kind of makes sense because there's very high levels of very long chain fatty acids, and the peroxisomes here are probably dysfunctional. And so what you then see is that if you compare this phenotype to the fly phenotype and you look at axons in flies, you see that in the normal circumstances, axons in flies are also insulated. And if you express this mutation and overexpress it, you see that this, just like in Mitchell, it creates a problem in insulation and creates phenotypes that are quite similar in fruit flies, where you see similar we can't call them demyelinating because there is no myelin in fruit flies, but it creates a problem in the glial cells of these flies. So we then turned to mice, and in collaboration with Hank Jung's Lee lab, did a culture of Schwann cells from mice. And Schwann cells are these cells that insulate these axons, and there is a lot of ACOX-1 in these Schwann cells, as is shown here. And the red marker is a marker for the Schwann cells. The green marker is the marker for the protein that we are talking about. And what you can see that if you overexpress in these cells, in these Schwann cells, this mutant protein that is found in Mitchell, you have a dramatic reduction of the number of Schwann cells. And this number of Schwann cells can actually be protected or restored partially by expressing a drug or treating the cells with a drug called NACA, or N-acetylcysteine. And so this provides a protection, and the reason is that this scavenges the reactive oxygen species that are produced by this constitutively active enzyme. And we then turned to flies and saw very similar data. So if you overexpress the Mitchell mutation, the flies are very short-lived. They typically live about 1 day. But if you treat them with N-acetylcysteine, 60% of the flies will eclose and live for a protected period of time, which is shown here. If you look at the survival, so a few of these animals that express this Mitchell syndrome mutation, you see that a few flies survive and they're short-lived. They live on about 25 days. If we feed these flies with this drug NACA, this that scavenges the reactive oxygen species, they do significantly better. We can also express catalase, which is the enzyme that normally scavenges the ROS, and these flies also do a lot better. So in a nutshell, I want to conclude that in Mitchell syndrome, there is an ACOX-1 gain of function mutation that makes a constitutive dimer that produces high levels of reactive oxygen species in cells where there is a lot of very long chain fatty acids. This leads to lipid peroxidation and leads to a loss of these cells. And NACA, which is produced by this company, Naquity, suppresses the reactive oxygen species and clearly improves the phenotype. I'm going to end with one other observation, and that is that this gene, when lost, so here I discussed the gain, but when it's lost, it leads to an elevation of the very long chain fatty acids. And that leads to a similar problem in these cells, but very different, because in this case, treating these animals with NACA doesn't work. We need to treat them with drugs like bezafibrate and fingolimod that will decrease the number of lipids or decrease the number of a product of these lipids, sphingosine 1-phosphate. And this is interesting because this protein leads to a neuroinflammation, and we currently using this model for a much more common disease, which is multiple sclerosis. So we've gone from a rare disease with Mitchell syndrome to a much more common disease, which is multiple sclerosis. Thank you.
Hi, I'm Michele. I am Mitchell's mom, and I want to say thank you to Dr. Bucelli and Dr. Bellen for giving you that clinical picture as well as the science researchy fruit fly stuff from Dr. Bellen. I'm just wanting to give you an update on what's happened and what we're doing kind of moving forward. So Dr. Bucelli fit 8 years into one slide, but I'm going to beat him because I have 12 years on this one slide. And I won't go through everything because Dr. Bucelli went through a lot of it, but just really a timeline of what's happened since Mitchell had his first symptoms through till today. And you already heard about most of those things from Dr. Bucelli. So when Mitchell passed away, a new NBC News reporter wrote this story, and I don't think at the time I really had any idea. I don't think any of us had any idea kind of what type of legacy he would leave. And there's so many different ways I could go with this. The research, the looking for treatments and cures. My entire job career changed because of Mitchell and because of my experience with him. I now work for the Undiagnosed Diseases Network Foundation, which came out of my personal experience as the mom of an undiagnosed patient. There are just so many different things that Mitchell and Mitchell's story have affected, and I'm so grateful for that, even though we miss him every day. One of the big things is my husband and I started a foundation called the Mitchell and Friends Foundation. You can find us on Facebook, and we also have a website. And really, it started because we wanted to help connect patients. As Dr. Bucelli and Dr. Bellen said, pretty quickly we started hearing about other patients around the world, and they would somehow find us, probably through the one published article. They would get Dr. Bellen's email, he would connect them with us if they wanted to, and then we would meet. And so the first thing we said is there needs to be a website that these patients can go to and actually find information. So my husband, Matt, did a lot of work creating a website, describing the disease in terms that a non-science person could understand. And then we started working to just build a community. We wanted people to not feel alone. We felt alone a lot during this journey, even though we had amazing doctors, amazing family, church family, friends, neighbors, so many people helping us during that 7-year period between symptoms starting and when Mitchell died. But it still just didn't feel the same as having another parent who could say, I know exactly where you've been. My child has Mitchell syndrome too. And so that was our main purpose when we started. The foundation was just to build connections, support patients and families. But as you heard, since then, we've moved on and are moving into the research area and fundraising and trying to get a treatment, because we ultimately realize that that's really what patients and families want. We had our very first Mitchell syndrome family meetup in the summer of 2023, and you can see that slide with the green outline on the left. We got to see the lab of Dr. Chung at Houston Methodist, who's working with the fruit flies still and working to see how riboflavin affects the flies. You can see one of our patients, Michael, who actually is holding a vial of fruit flies that have the exact Mitchell syndrome mutation that Michael has. And it was really inspiring, I think, both for Michael and his family to see the lab, but then also for the people working at the lab to meet Michael and meet his family and really see what it is that they're working for. These other photos are patients who've come to St. Louis to have a clinical visit, either with Dr. Bucelli, or we have a pediatric neurologist, neuromuscle specialist, Dr. Renata Knox, who's seeing our pediatric patients. And when they come, we do our best to meet them and help to take them to their appointments, answer questions, and just, again, really trying to build that community so they don't feel alone. We recently had a gathering here in St. Louis. We were able to donate more money to the Department of Neurology, and we had 3 families from all over the United States, patients and families, come visit the lab. Dr. Hubler gave a tour of all the work they're doing on the ASOs. Dr. Miller, Dr. Bucelli, everyone else in the department just was so gracious. And I think, again, it went both ways. The patients and the families were excited and inspired and filled with hope, seeing all the work being done. And the team that's working at WashU, I think, we're also inspired and challenged to keep working, meeting the patients and meeting the families and seeing them in the flesh, in real life. Finally, this is just another example of connecting. My daughter Miranda is the youngest of 3. And you can see that picture in the middle. She's with one of our patients, Augie, who she just really loves. And then on the far right, all of those girls there in that picture, they are all sisters of brothers with Mitchell syndrome. So forever, Miranda felt like she was the only one. It's just me. And now she has a whole girl tribe of sisters who all know what it's like to have a brother who is struggling and going through kind of a journey that really is, difficult for families and definitely difficult for siblings. We've learned a lot about the challenges that siblings face. And then at the bottom there, you can see two of our patients, Michael and Graham. They were able to connect, and it's just been a really great time getting to meet everyone in person and provide that support. So there's more information on our website, mitchellandfriends.org. Like I mentioned, I do work at the Undiagnosed Diseases Network Foundation, and my email is there. It's Michele with just one L. I'm happy to answer any questions about Mitchell syndrome, or if you yourself or your child or loved one are on a diagnostic odyssey, I'm happy to help as much as I can. Thank you so much.
Our undiagnosed case study will be presented by Dr. Kathleen Sullivan and Erica Shindewolf. Dr. Kathleen Sullivan is the Division Chief of Allergy Immunology, Professor of Pediatrics, and the Director of the Undiagnosed Disease Program at the Children's Hospital of Philadelphia. She trained at UCSF and Johns Hopkins and has been at the Children's Hospital of Philadelphia since 1993. Her clinical interests include the study of patients with DiGeorge syndrome, chromosome 22q11.2 deletion syndrome, variables related to the outcomes of patients with primary immunodeficiencies, and developing diagnostic methods using advanced technologies. Erica Shindewolf is a licensed certified genetic counselor specialist currently serving the Undiagnosed Disease Program at the Children's Hospital of Philadelphia. Erica is the Leadership and Education of Neurodevelopmental Disabilities, also known as LEN, Genetic Counseling Director, as well as the co-director of the Reproductive and Developmental Genetics course and Professional Development courses at the University of Pennsylvania Masters of Genetic Counseling program. Erica is a fierce advocate for trauma-informed care and is committed to creating better care models in genetic counseling for families undergoing a diagnostic journey.
Hi, my name is Kate Sullivan, and I'm here today with Erica Shindewolf. We're going to talk about an undiagnosed case study today, and I think it exemplifies some of the technical problems, some of the frustrations, some of the strategic pathways that are being developed to try and solve these unsolved cases. So I'm going to start with the patient because that's where everything starts. This is the team that worked on this case. You can see, well, first of all, it's still under investigation because it's not solved. You can see this is a lot of people. We are really proud of our team, and everyone has come together for this family. In spite of being unsolved, we do think that there are a few opportunities to still make progress, and I will highlight those as we move forward. So this is the family. You have heard from Christine McGarvey. She is in the middle here. So this is Christine, and she has some medical issues that she has been living with for quite some time. These are all what we would consider to be autonomic problems, meaning the autonomic nervous system appears to be impacted. Now, you never really want to make assumptions, but if I had to sort of tie this together with a theme, that would be the theme that I would highlight for Christine. Her daughter, Bridget, is actually the proband in the UDN study. And Bridget has less severe issues, but she's, of course, much younger. She could potentially evolve additional problems. We sincerely hope she doesn't. But at the moment she was enrolled, she had bowel and bladder dysfunction. She had dysfunctional voiding. I'll talk a little bit about that in a minute. Had alternating constipation and fecal incontinence. And she had some mild developmental delay, but had really had a lot of catch up by the time we met her. Now, there's really other family members that you need to meet because they are part of the analysis. So Christine has a sister who's over here. This sister has hearing loss and urinary incontinence, but also has a bladder malformation. So it's not 100% clear that it's exactly the same mechanism. And she is factor 11 deficient, which you will notice is also true for the parents. Patricia, the grandmother to Bridget and the mother to Christine, also is factor 11 deficient. She had a very mild form of PKU that was managed with diet. But the reason I'm highlighting her now is, although she did have some medical problems in her younger years, in her 60s, she had quite a precipitous decline, is currently hospitalized and not doing well, characterized by ataxia, neuropathy, chorea, altered mental state, and renal failure. Again, there's an echo of the clinical picture that we see in Christine, but not precisely the same. And then Christine's father, Bridget's grandfather, has hearing loss, factor 11 deficiency. And then he has a sister with gastroparesis and celiac disease. So this is a complicated family history. I think our initial assumption was that there was an autosomal dominant condition with female transmission. We are less sure of that now, and I'll explain why. And of course, we have analyzed it in various ways, which you will see in a minute. So these complicated stories are actually one of the hallmarks of the UDN and the UDNF. If it was an easy case to solve, it would have been solved before they got here. But this does introduce a complexity to the analysis, and I'm going to spend a little bit of time highlighting that. So, as I mentioned, Bridget was our proband. So the little girl was the official enrollee. And at the time of her enrollment, her major issue was voiding dysfunction. When we saw her, she'd actually been studied multiple times, and she had the staccato voiding pattern. That might not mean anything to you, but when you void urine, you need the sphincter to open and the bladder to contract, which is the exact opposite maneuver. And there's a neurologic circuit to ensure that they are opening and compressing at the same time. And so when you see the staccato voiding pattern, the bladder is contracting against a closed sphincter. There's a mismatch between what the muscles are supposed to be doing. And there are a very broad range of etiologies that can lead to that picture. But it was a very clear clinical phenotype in Bridget. I mentioned that she had some mild developmental delay, but had had significant catch-up. And the other thing I wanted to highlight is that she did have this additional flavor of autonomic dysfunction in the sense that she had significant constipation, but would also sometimes have a little bit of smearing in her underwear. She did have some mild fecal incontinence, which is sort of the colon version of the staccato voiding pattern that we saw. So it felt very aligned with her clinical picture and certainly can be part of an autonomic dysfunction syndrome. Now, whenever you see a female dominant inheritance pattern, you always want to think about mitochondrial disease. And this picture could be consistent with mitochondrial disease. You probably didn't notice on the slide of our team, but we do have a couple of mitochondrial experts, and Bridget was seen by them. The typical screening tests are lactate and pyruvate and GDF-15, which are imperfect screening tests, but they did do a thorough clinical evaluation and did not think that this was likely to be a mitochondrial disease. Now, Bridget's symptoms were not particularly alarming, although certainly problematic. I don't want to underestimate their impact on the family. But in this case, the family history was a really compelling driver for the workup. Because there was at least an echo of the same clinical features across 3 generations, we really wanted to be sure we were ruling out as best we could a genetic contributor to this picture. And so Bridget's our proband, but you always have to put it in the family context. Now, Bridget had had some genetics, as had the whole family, before their first visit to our UDN site. And she was known to have compound heterozygous variants in a gene called ZBTB40. The ZBTB family is a large family and one that's known to most geneticists. This is not a disease. This is not a gene that's known for sure to cause disease. Many of the ZBTB gene family are associated with human disease, but this one's not. There have been 2 reports of children with autism and developmental delay that have mutations in ZBTB40. So it's 2 kindreds, it's not a lot to go on. Here's why it's a little confusing. In the mouse model, the mouse phenotype is all about the bones. So not really anything about development or behavior. The mouse model is all about bone density, and the males are infertile. So that's a little bit hard to align with this family. And then in vitro, surprisingly, this gene has an impact on telomeres. Telomeres are the ends of the chromosomes. And when telomeres are dysfunctional, there's a very specific clinical phenotype, which is not aligned with Bridget, Christine, or Patricia. Possibly with Patricia, but certainly not aligned with Christine or Bridget. So they were enrolled in the UDN, and we did whole genome sequencing as our first effort. The clinical report that we get from Baylor was negative, but they do provide a research report. That research report again identified the ZBDB40 compound heterozygous variants. Another gene was identified, ZNF704, that had compound heterozygous variants. This again is not known to be causal for human disease. It's thought to be involved in circadian rhythm based on mice studies. Again, doesn't really align with the family. We always pay attention to de novo variants, although with this family history, we didn't put quite as much weight on that, but you really don't want to ignore a de novo variant if you can. De novo just means it's new in that patient. And remember, this is focusing on Bridget. So VGLL3 is known to be associated with autoimmunity in a murine model where they overexpress it. There actually hasn't been a model where it's been knocked out or knocked down. And that brings us to the last one that I've highlighted in red, which we were initially very excited about. So this is a receptor for a neurotransmitter, and this receptor had compound heterozygous variants, meaning one variant inherited from mom, one variant inherited from dad. And why is this relevant? The knockout mice, which would be roughly the equivalent of Bridget in this story, have fewer myoenteric ganglion cells. These are the cells that tell the muscles to squeeze and are important for coordination of that muscle squeezing. So this actually feels very aligned with Bridget, but the knockout mice would represent an autosomal recessive inheritance. And of course, the mother would be heterozygous, and we don't know about the grandmother, Patricia, who had the later neurologic decline in her life. So this is quite problematic in terms of ascribing this to Bridget. And so when this happens, which happens all the time in the UDN, you have to sort of go back to basics. And so we check our assumptions. Do all three members have the same thing? I've tried to highlight some differences as I've gone along. Christine appears to have a more severe disease than Bridget, and whether that's age dependent or whether it's truly something different, we don't know. And then Patricia looks different yet in the sense that she evolved her current neurologic condition after the age of 60. So that when we run into trouble, we always have to challenge our assumptions. That would be the first thing we would think about. Could this be polygenic and autoimmune? So certainly there are lots of multiplex kindreds for arthritis, for psoriasis, for inflammatory bowel disease, but it's polygenic. It's not one gene causing the disease. That's common for autoimmune conditions. But we did our due diligence. We looked for autoimmune causes of dysautonomia and did not find evidence of that. Always worth asking, could this be environmental? Could it be something completely wacky like arsenic exposure? So families that live in the same house could conceivably have the same environmental exposures. We didn't have any evidence of that, did not pursue it extensively. There wasn't a clear toxin phenotype that aligned with their clinical picture. And then it's always time to ask, is this a new gene or a new phenotype of an old gene? So we have gone through this exercise multiple times for this family because it is, I think, a frustration to all of us that we have not been able to find an answer. So I'm going to turn this over to Erica Shindewolf, who's going to talk about what do we do in this circumstance. It's a problem that we all face in the UDN. What's our path forward?
Thank you so much, Dr. Sullivan. Just as Dr. Sullivan kind of reported here with Christine and Bridget's case, there's a lot of weight and power in genomics. And as you can see, there are so many layers of complexity when it comes to interpreting these results. And I want to take our conversation in a direction to talk about why that's so challenging and to discuss the limitations of genetic testing, and then to reiterate what Dr. Sullivan was saying about how do we move forward and kind of where do we go. So why is this so challenging? What are our pitfalls and kind of where do we go to solve these questions for families? Two of the limitations that I want to talk about first, and you'll see with the second end of this talk, we're going to get a little in the weeds with the technology and genetics, so just bear with me. But we're going to first talk about the complexity of looking at two different types of variants, regulatory and structural. Genetic testing does have limitations, as we know, as we're seeing with this family. When you really think this has to be genetic, And yet maybe we don't have the knowledge or the technology at that point to figure it out. The first thing to think about with regulatory regions is that there is an inability to detect all regulatory variants in genetic testing. And what regulatory variants are variants that affect gene expression rather than the gene sequence itself. These variants can kind of hide in promoter regions or enhancers within the genetic code or other regulatory elements. And so they can be really difficult to identify in the kind of standard genetic testing that we do, because they don't necessarily change the protein coding sequence of a gene, but instead influence how and when a gene is turned on or turned off. And so we might not be able to find them with the technology that we have. We also may not be able to understand the relationship between regulatory variants and disease. And this could be because we lack comprehensive databases, and we also lack the functional context to really draw the necessary conclusions to say, this regulatory variant that maybe turns off a gene could potentially lead to this disease. We're just not there yet in our understanding, but we're certainly working to get there. There are also challenges with structural variants. Structural variants can be really hard to detect. And so structural variants are usually areas where maybe there's missing pieces of the gene or extra pieces of a gene. And sometimes they can be too big or too small to look at in the way that we typically do genetic sequencing or assessment of our genes. Their clinical significance can be really challenging as well. So the effects of structural variants are really not always straightforward, and the relationship with disease can either be indirect or involve kind of the complex interactions that Dr. Sullivan was talking about, you know, their interactions with other genes or their interactions with the environment as well. There's also gaps in the reference genome. So when we're looking at structural variants, we're oftentimes comparing the person or the patient that we're thinking about to a reference genome. And much of genetic testing really relies on that comparison, but that comparison may not be fully representative of genetic diversity in different populations. Variants that are specific to certain populations or those that might occur outside of regions that reference genomes miss may represent gaps that we just can't address until we do more genetic testing for more people and kind of get a better understanding. There's also an incomplete understanding of functional mechanisms with both structural and regulatory variants in disease. And Dr. Sullivan kind of already hit on this, but really the role of these variants are not always well understood. Their effects can be really subtle, or they can involve kind of multiple steps in gene regulation and expression. Even when these variants are detected, predicting their functional impact on disease pathways is not always straightforward. And many diseases, particularly those that are complex or progressive over time, can really be influenced by a combination of structural and regulatory variants within the gene, alongside environmental and lifestyle factors, kind of being this multifactorial effect. And detangling these influences can be really challenging, which leads to challenges in providing kind of a clear, actionable answer for patients. Now, all that to be said, we're going to move forward and think about kind of what are the strategies to enhance existing genetic analysis. And again, there are many different ways in which we're trying to fix these problems and really look at the genetic information in different ways. The first that I'm going to talk about is this idea of long-read DNA and RNA. And to talk about that, I like to use an analogy of thinking about reading a book. And so the way that we've thought or traditionally done genetic testing is through something called short read sequencing. And short read sequencing is sort of like opening a book and reading individual words on a page, or maybe short phrases from different pages, and trying to get the context of the book that you're reading. And you can imagine that if you're only getting a few words or you're only getting a few sentences, you may be able to piece some things together, but you're certainly not getting the full level of comprehension that we're used to. And it's amazing to think about that, because even doing short read sequencing is such a huge jump from where we were 10 years ago, but that's the level of genetic kind of technology that we have, is kind of looking at these phrases, looking at these sentences, and trying to put all of the pieces together. Long read sequencing is a little bit different. So instead of reading individual words or short phrases, now we're reading the entire sentence or the entire paragraph or the entire chapter of a book. There's way more context, and you can understand how these individual words and ideas connect together. You get more of the context. And so we're moving in genetic technology to being able to get way more of that context. It's just much harder to do that. Long read sequencing, instead of taking 100 to 300 base pairs of genetic information, takes thousands of base pairs of genetic information and really sequences to look at those spelling differences to see if there are differences or mutations that lead to alterations in the DNA that could cause disease. So that's one thing that a lot of folks who are in the UDN are moving towards for families who currently don't have answers, really being able to look at that in much more detail. RNA sequencing can be done, again, short or long read. And RNA is a little bit different because it really looks at gene expression. So it's looking to see, it's really sequencing the RNA molecules within the cell. And that's just seeing if the gene products are turned on. It shows which genes are active, how much they're being expressed, and the abnormalities in gene expression that may cause disease. If we see mutations that affect gene expression or how RNA is processed or how a gene or protein is processed, that can actually help tell us a lot of if that mutation is meaningful. Is it doing something to the way that the body is reading this information or reacting to this information? So it gives really important information on the function of genes and helping to link those DNA changes to different diseases or traits. Splice analysis is similar but different. And again, like I said, we're getting pretty technical. But splice analysis looks at different genetic changes in the process of splicing. And splicing is really removing non-coding regions of an RNA when we're making the final messenger RNA to make proteins. Splice analysis helps detect splicing errors that are caused by mutations. And a lot of diseases are caused by splicing errors or disrupt gene function because it splices the gene and it doesn't allow that gene to be processed the way that it's supposed to. So by looking at splice analysis, we're often able to see, if there's a functional change that can be helpful in understanding whether or not this mutation contributes to disease or to a greater disorder. Somatic variant assessment is a little bit different. So a lot of the analyses that we were talking about really look at the germline, look at kind of the information in the DNA that you were born with, that was inherited by your parents. Somatic variants can be a little bit different because they're genetic changes that occur in the cells of the body that are not coming from egg or sperm, but that can change over time as we grow or even just after we become an embryo and start to divide. Unlike inherited variants, somatic variants are not often passed on to the next generation. And somatic variants are really common in some types of conditions like cancer. A lot of times cancer is the result of a somatic change or a cell-specific change that then replicates and causes a ripple effect in someone's body. There are other genetic conditions that happen in the same way. And so we can look at single cells by using different types of somatic variant assessment to really look at highly detailed areas of that cell to see if there's potentially a difference in a single cell or an affected area of somebody's body versus their germline genetics. And those differences can really help us because sometimes we're just not looking at the right source. So sometimes if we're looking at blood, but we really need to be looking at an affected tissue, or we're looking at saliva, which is a different cell line than your blood, it can change the way that we see how our DNA is processed or what the changes of the DNA are. And that can be really helpful when we're trying to find these really rare variants. All of that to say, when we're combining these strategies, we're really helping and hoping to gain a clearer and more complete picture of genetic variants and how they contribute to disease, to hopefully, of course, lead to better diagnosis, understanding, and even potential treatment. But we have a long way to get there before this kind of technology becomes clinically available and affordable and accessible to a lot of families. We know, and certainly people who are tuning into this know, that the diagnostic odyssey which is the journey, of course, that we talk about for undiagnosed families, is frustrating. It is riddled with an emotionally taxing process. It can take years or decades, and it's marked by certain uncertainty, frustration, and even at times isolation. There's so much that needs to be done to support these families. And, you know, organizations like the UDNF are certainly a way to do so, but it is the goal of clinicians who are taking care of these families to really help support families in dealing with this very long journey, whether that be by, you know, managing expectation, figuring out preferences of how you want to learn information, where you want to be at this process, what does regular check-ins look like, making sure that families have appropriate psychosocial support connection to other undiagnosed families or families that have similar, you know, processes or similar things going on. So important just to make sure that, you know, when you don't have a unifying diagnosis, it can be really hard to get services. It can also be really hard to just feel like anyone understands what you're going through. And so there should be a different level of psychosocial training that is coming to clinicians who are taking care of these families. But at minimum, resources that are available to these families, such as connecting with support groups, to know that these families are not alone. And certainly efforts like the Undiagnosed Disease Network is a way to get people involved and connected with clinicians who are not going to give up on this journey or going to walk alongside families who are still wanting this diagnosis and able to weather the storm of uncertainty with us.
So I think we'll just summarize by saying it's been our great privilege to collaborate with Christine and her family and go through this process together. She's an inspiration to all of us.
Thank you so much, Dr. Sullivan and Erica Shindewolf, for discussing our family's case. If I could give a title for our family's diagnostic odyssey and my experience as a UDN participant, I would borrow from Walt Whitman and entitle it, "Oh Captain, My Captain." For the longest time, I felt like I was the captain in a rowboat during a Category 5 hurricane, and I was constantly trying to bail water. Now the UDN is my captain, navigating the uncharted waters of living with an undiagnosed disease. Being a participant in the UDN has truly been life-changing and transformative for me. It has been life-changing in the fact that I now have peace in knowing that I've done everything possible to help both me and my daughter, regardless of whether or not we receive a diagnosis. The UDN has also helped me navigate the unexpected turn of events when my mom, who also was my caregiver, suddenly became ill with an undiagnosed disease. I am truly grateful for the UDN for helping me navigate this, as I am her only living family member, and I was completely overwhelmed. I'm also incredibly grateful and in awe of all the cutting-edge research techniques that are being used to diagnose patients. Being a participant also has been transformative in the fact that has given me the confidence to be my own advocate for myself and for others. I had the confidence to become a rare disease advocate with the National Organization of Rare Disorders and as a UDNF peer member. Becoming a rare disease advocate has helped me make meaning of living with an undiagnosed disease and all the challenges and sufferings that come with living with the undiagnosed disease. And is the most truly the most fulfilling thing that I have done so far in my life. In addition, I have gained a community of people who understand the challenges of living with an undiagnosed disease through my participation in the peer group. I also feel less isolated and alone because I know that there is a whole team of scientists and clinicians working to solve medical mysteries like ours. Also, as a peer member, I have seen firsthand the dedication and commitment of the PIs and researchers to solve undiagnosed cases. In conclusion, I would like to quote a famous cricket player and an actor, S. Srisanth, who said, "A good captain is great only if he has a great team." And that's what I have gained through my participation in the Undiagnosed Disease Network. Dr. Sullivan is my captain, and she has the whole network behind her as her team, working on our case and other families' cases within the network. Our next speaker is Dr. Matthew Might, and he will be discussing advances in research and therapeutics. Dr. Matt Might is the director of the Hugh Cole Precision Medicine Institute at the UAB and a rare disease dad whose son Bertrand was the first documented case of NGLY1 deficiency.
It's really a pleasure to be here today and to be able to share with you all some advances in research that I think are very relevant to this community in particular. That said, before we get started, I have to issue a kind of standard disclaimer. I am actually required by UAB to give this disclaimer, which is to say that if there's anything that looks like medical advice in this talk, you really should not be taking it. And that's because I don't actually have any medical training, and I like to give medical advice. Or really, I like to teach computers how to give medical advice. So if you spot anything that looks like medical advice in this talk, consult a physician before following that. So really, there's just one big idea I want to get across today, which is it is now becoming possible to do precision therapeutics in practice. And I'll define, what this means in just a moment. But I also want to get into the two big advances that are really driving this. What is it that is finally making these individualized precision therapeutics a reality? To spoil them, it's really a lot of AI and CRISPR. So there's both, computational technologies, but also some biological technologies that are making this possible now. And I do want to share my perspective, the way that I approach this problem, which is to say that I really have multiple hats when it comes to rare and undiagnosed disease. For me, it all really began with my son, Bertrand. So I was actually out as a professor in computer science, just starting out my professional career when he was beginning a diagnostic odyssey. I think in this picture, he's about 1.5 years old, which is in the earlier phase of that odyssey. And it unfolded over the course of about 4 years. And it ended with, one of the first applications of exome sequencing as part of a clinical research study at Duke University, now one of the UDN sites. And he also ended up being the very first case ever discovered of his particular genetic disorder. So sort of a double first in that sense. And that really launched me into another odyssey, which is the major point of today's talk, which is the therapeutic odyssey, which I really spent the rest of his life pursuing, which is, okay, if you know exactly at a molecular level what's wrong, what do you do about that? How do you find treatments that can actually make a difference? And if you fast forward through a lot of other stuff in my life, you land into my current job, which gives me the perspective I bring today, which is as the director of the Hugh Cole Precision Medicine Institute at UAB, where our goal really is to try to do what we think of as, science in the service of patients. And I guess I should clarify, totally that I really am just a computer scientist that has flipped over into medicine, despite the fact that I run this institute. And when I say science in the service of patients, I really do mean trying to, find science that stands in the place of standard of care when there is no standard of care. So I view science as the thing you do when you've run out of medicine to do. We have a fabulous medical leadership team that helps run this program. I also like to give credit to a team of undergraduates that also helps that medical leadership team when we reach out and engage with patients. I think of them, or somebody wants to describe them as the care traffic controllers, in the sense that they engage with patients and physicians, clinicians, and then computational tools in order to find things that can help patients. Before we dive into any specific way that you can do this, I want to talk about what precision medicine as a whole really is. And if you go back to the beginning, there's this report from the National Academies. It's from 2011, and it's trying to define really for the first time this emerging field, this emerging practice of precision medicine. And in the glossary, there's a very long and somewhat confused definition of this whole field. But if you really boil it down, what it really is precision medicine is what happens when you're really tailoring medical treatment to the individual characteristics of each patient. So it is somewhat synonymous with personalized medicine. And what's interesting about this report is that the word data appears on almost every page. And really, it's patient data, very specifically in almost every case. And what that means to me is that when I think about operationalizing precision medicine, I think of it as a process. And what we're really trying to do is optimize health with data and patient data in particular. So the way this process looks is, you've got all the data around a patient and you're trying to find the set of potential or best possible treatments for them. So I think if you look at the kinds of data we're talking about, the piece of data that got everyone really excited about precision medicine in the first place was the clinical accessibility of the human genome. Once this became available, it was clear that we were going to have to use some computation in order to start doing medicine, because the genome is so big that you really have no choice but to resort to computational methods to deal with it. But there's always been lots of data out there on patients. If you just look at all the longitudinal data available in the health record, I still don't think we've done a very good job of exploiting all of that in terms of optimizing patient health. And then, of course, there's all the data outside of patients. There's all the knowledge in books, papers, manuscripts, biomedical data sets. And if you think about it, all of this, all that knowledge, all that data should always be sort of used together when you're thinking about how to find the right treatment for a patient. But this is in some sense, for a single human mind, this is an intractably difficult problem. No human's read every paper, no human can hold all the data on a patient in their head. And so precision medicine in a lot of ways is really about utilizing computation very effectively inside medicine. And so we'll talk briefly about that, the kinds of computation that can help patients find therapies today. So if you really, look at this process from a very high level, where you're going from a patient over to potential treatments, it kind of, breaks apart into two phases. There's this precision diagnostics phase, and there's a precision therapeutics phase. And to a certain extent, the UDN is the gold standard for doing precision diagnostics. Now, there's literally no entity on the planet better at this task than going from a patient over to a root underlying molecular cause of disease or other cause of disease, if it's environmental, for example. And then, after that, comes, I think, the most interesting part, which is, okay, we've got a diagnosis, but now what do we do? It's good, it's powerful to have a diagnosis, but I think there's a yearning in every patient or parent's mind to want to know, what else can I do now? And that's where I've spent a lot of my professional career since, is figuring out how to answer this question. So when you ask, hey, how do you do precision therapeutics? I think there's really sort of two broad ways to do this. The first is what I would call the rational approach to precision therapeutics, where you've got your hands on the root molecular cause of disease, some mechanism of harm. And now you're trying to figure, okay, well, we know it's a variant in this gene that's driving the disease. That's the mechanism. But what is it really doing? Has it made that gene overactive, in which case we need to find an inhibitor for it? Has it made it underactive, in which case we need to find an activator for it? Has, a combination of mutations perhaps wiped out the activity in a gene, so the activity is absent now, and now we need to somehow compensate for that absence or replace its original activity? Or is it the case that this mutation actually somehow shifted, the gene product itself, the protein, so that it's now toxic in some way? And now what we have to do is eliminate that toxic gene product. So these are the sort of four broad directions you can go when you look at a mutation and you ask the question, what should I do about it? Now, there's another way to approach precision therapeutics that doesn't ask that question. And in this approach, we call the empirical approach. you take the knowledge of that mutation. So you do know what's driving the disease, and then you build some kind of biological model or a biological avatar. It could be patient cells, it could be genetically modified patient cells, it could be a model organism like a fly or a mouse, and then you start testing things on the model. So in this case, you might not know exactly what that mutation has done beyond the fact that it is in fact the one responsible for the disease. And then you just see, can we correct it in the model? So these two approaches they can actually be quite complementary. There's no reason you can't do both at the same time. And both of them have really kind of been enabled by advances in technology recently. And again, those two advances really are artificial intelligence on the computational side, driving improvements in this rational approach to precision therapeutics, and then technologies like CRISPR for genomic engineering on that wet lab biological side. And that's really what is making it possible in a lot of cases to build these precision disease models in the first place. So I'll talk briefly about both these approaches, and I'll also talk about some of the newer advances in AI and the implications they have for precision therapeutics. I want to start by talking about a classical AI system that we built and use all the time at the Precision Medicine Institute at UAB, something that's been very generously funded by NCATS at NIH. It's a system we call Medicanron. In a nutshell, here's what it is. And I think it's important to understand, at least at a high level, what it is. You can potentially understand whether or not this kind of system might be relevant for you. It's really a fusion of logical reasoning and all the biomedical knowledge that we can get our hands on. So by all that we can get our hands on, at this point in time, I think it's around 180 some biomedical data sets. And that includes, a condensation of the entire biomedical literature as just one of those data sets. So, at a very high level, here's what you get when you put all this together. What you get is old school logic combined with high-speed automated reasoning to achieve superhuman-like deduction with, we hope, clinical insight. I think the clinical insight part is one of the most challenging. That's why we have really talented medical directors to help us work with the output of these tools to make sure that we're actually providing meaningful insights for patients. And I think it's useful to look at what's inside this thing. Like, how does it think about the world? Inside of its head, you'll find what's called a knowledge graph. And a knowledge graph is essentially a collection of circles, for lack of a better way to describe this. And every circle in its brain is actually a noun. In this case, it's a noun from the biomedical literature. So it could be a gene, a drug, a disease, a phenotype, something like that. It's a concept, it's a noun, it's a thing. And then what you'll also find are arrows drawn between these circles. And each arrow represents a verb. So you have a noun, a verb, and a noun. You have a subject, a verb, and an object, which means you have a sentence. Every time you draw an arrow between two nouns in this AI's brain, you've just added a sentence to its knowledge base. But you've done so in a very structured way. So for example, you might find a sentence like this lurking inside. In fact, I know you'll find this sentence lurking inside its brain. Prevacid inhibits NGAze, just represented by this arrow from Prevacid to NGAze and labeled with inhibits. And the reason it's important to have this kind of structure is it enables logical reasoning. So for example, if we know that concept blue, whatever that is, increases concept red, whatever that is, and we also know that concept red decreases concept green, we can logically infer something, that by increasing the decreaser of something, we should ultimately decrease that thing. So we can infer that blue should decrease green. So that's a we wouldn't say that it's absolutely certain because biology can do strange things, but now we have a working hypothesis that blue decreases green. And we can use that potentially when we're trying to work with the goal of finding a precision therapeutic. So its brain at the end of the day is a soup of these circles and arrows stitched together to the tune of, I think, billions of these arrows at this point. And we can search through it. Or really this AI can search through it. And we can ask it questions like, if we're working with a very specific low-level target or a specific gene, we can say, okay, can you find some pattern in this graph that enables us to predict what will inhibit or activate this target? Because that 75% of the time, I think that's the question we're asking, is we know the specific gene we want to throttle up or throttle down in some way. And so we're just looking for ways to do that. So we can ask that question. And I'll just give you a very quick example of what this can look like in practice. This is an actual case that we worked with some years ago. It's one of the first ones we actually worked with for Medicanron. This amazing mom here reached out to us. She runs the ADMP Foundation, Sandra Sermoni, and she said, "Hey, I heard you're building an AI that can potentially predict things that can help." And so the question was, what activates ADMP? It's known in that case that the disorder is really driven by a haploinsufficiency or an insufficiency of activity in this particular gene. The gene's not gone, it's just underactive. So the question is, how do you activate it? And it came back very quickly, a fraction of a second, and looked at the cancer literature. In fact, if you look at how it figured this out, that it seems to be the case that low-dose ketamine should upregulate the activity of this gene. And so took all this information back to the foundation. They chewed on it for a while and eventually felt confident enough to, fund a clinical trial. That clinical trial launched, and they actually got it completed, I think, around summer 2021. So it took about a year. And bear in mind, this is all during the middle of COVID, and they still actually pulled all this off. And sure enough, some of the core predictions held up. We did see a doubling of, the expression of this gene, which was what we wanted to see. But more than that, we actually saw really significant clinical improvement for the patients, which is what we really, really wanted to see. And if you want to know more about this from the sort of the patient perspective and how all this began with a little bit of a suggestion from AI, you can read about it on STAT. It was published scientifically not long after that. And if you want to know the sort of details of the trial itself, you can read about it here. But I do want to talk about that other approach to precision therapeutics that I mentioned, that the empirical approach. And, in contrast to artificial intelligence, you might think of this as organic intelligence. It's actually a phrase I've stolen from Ethan Perlstein, who's also a believer in this particular approach. We actually do have, another center that I help co-direct at UAB for this, called the Center for Precision Animal Modeling. And Brad Yoder and I actually co-direct this center. We have a very large faculty actually that work together very collaboratively on this to make it work. And really what it does is, again, we start off with the known or suspected genotype of a patient, and we use that to develop a precision disease model. And it can be in cells, it can be a zebrafish, it can be, xenopus or tadpoles, it can be a worm, it can be a mouse or a rat. These are the kinds of models we can build. And then, once we've got the model, we check to see, okay, is there some signature of disease in this model? Is there something in this model that's abnormal now that's telling us about, this disorder, or something we can look for to change if we try treatments on it? And so, this is a thing we've done several times now. And so Kami Crowder, one of our junior faculty in the Institute, she built a zebrafish model for a disorder driven by mutations in the gene MAPK8IP3, and she tested some of the predicted therapies that came out of Medicanron on these zebrafish. And what she found, amazingly enough, a couple summers ago, was that amantadine was rescuing some of the aberrant behaviors she was seeing in the fish that had this, essentially humanized version, or the zebrafish equivalent of this genetic disorder. So this was a remarkable finding. She ultimately relayed that to the team working on this condition, and to my understanding, has even made a difference in the lives of some patients that have this particular disorder. So I want to close by talking about a recent shift in technology, a major advance that goes by the name generative AI. So what I showed you with Medicanron was a great example of a classical AI. It's classical in the sense, or symbolic, because it represents knowledge in a very symbolic way. Generative AIs actually have brains that work more like what we have. I'm not going to go into the details of how to build an artificial neural network or things like that. I'll just give you a broad definition that a generative AI is one that has sort of human level creativity. And that includes creativity in the sense of how we have scientific creativity, and we can hypothesize interesting and potential directions to go for patients. The best example of a generative AI that people are probably familiar with is ChatGPT. And the neat thing about generative AI, and this, when it's a linguistic AI like this, is you can actually ask it to explain itself. You can say, explain to me what generative AI is, and it will actually write back to you and give you a coherent explanation of what generative AI is. And there's lots you can do, and lots we're doing, certainly with these linguistic generative AIs, from the perspective of precision therapeutics. But I think in terms of finding interesting potential therapies for rare genetic diseases, while these will certainly help, I think there's other lesser well-known generative AIs coming into being that I think will make a much bigger difference. So for example, there are generative AIs, Google's AlphaFold is an example of one of these, that can predict the three-dimensional structure of a protein based on no more than its DNA sequence. So that's an incredibly powerful capability. Not long ago, and not long ago meaning like 5-6 years ago, when you wanted to know the 3D structure of a protein, you actually had to get X-ray crystallography involved or cryo-EM or these really expensive experiments. And now you can just ask a computer to say, well, here's the genetic sequence. Tell me its structure. So, for example, if you start off with the sequence of amino acids in a protein, it literally can transmute this directly into the 3D structure of that protein now. Again, just an unthinkable technology just a few years ago. With that as a foundation, you can start building lots of other interesting things on top. Something I sometimes call it generative medicinal chemistry. I've got a collaboration with Michele Dumontier at Maastricht University where we're extending this to go even further, where you can go from DNA all the way over to predicted small molecule therapies that could help. For example, if you're starting off with a protein with a mutation in it, can you go all the way to a small molecule that might help address the effects of that mutation? And the remarkable answer seems to be like, yes, this is likely something we're going to be able to do. In fact, Michele's already built some very basic working prototypes that do exactly this. So it's a very, very promising emerging field right now. And just want to close by reminding you that while there was medical advice in this talk, I'm not qualified to give any of it. So please consult a physician before you even consider it. And with that, I very much appreciate your time and attention.
Our last speaker is Dr. Meghan Halley, who will be concluding our webinar and will be discussing the importance of ongoing research. Dr. Meghan Halley is an assistant professor of pediatrics in the Center for Biomedical Ethics at the Stanford University. A medical anthropologist by training, her research focuses on ethical and policy issues arising from the introduction of new genomic technologies for diagnosis and treatment of rare diseases. She became interested in rare diseases after her second child was born with multiple rare conditions and was enrolled in the Undiagnosed Disease Network. She is a founding board member and current chair of the UDNF.
Hello, everyone. My name is Meghan Halley, and I have the great honor and pleasure of chairing the board of the Undiagnosed Diseases Network Foundation. To close this incredible event, I'd like to thank our presenters, the patients, the families, the clinicians, and the scientists who have made this such an insightful and rewarding presentation. And I also want to thank our UDNF staff, particularly Sarah Gentry, Dana Sayer, and Carrie Borello for their hard work in pulling this together. Every time I get to see and hear from the wonderful groups that work together so closely through the UDN and the UDNF, I am truly encouraged that the science of not just the future, but of now, is capable of finding a diagnosis for every undiagnosed patient, and also moving towards therapies and other supports to ensure that these patients and their families have the best quality of life possible. If you enjoyed this event, I hope you'll consider joining us in the next year. We have a lot of things we're planning underway, and you can find out about all of that as it comes up on our website at udnf.org, where you can also sign up for our newsletter so you can have announcements come right to your inbox. And finally, to wrap up, if you enjoyed this if you enjoyed this webinar and you'd like to hear more and support this incredible work, we would appreciate if you would please consider contributing to these efforts in your year-end giving. We truly, truly appreciate your support. Thank you very much.